Yeah, I know that all the time.
Didn't he tell us if we did that, he'd like take points from? He told us that people would do that and we
Well,
don't.
he did it, everyone did it last year.
Maybe it's
The only ones that you didn't turn it in, and the ones that turned it.
Yeah.
He turned it in. I don't need a feature.
Oh, there's like four of them, the first one is like an interval.
Okay.
Figuring out the.
So hopefully we're ready for the day, ready for Friday to begin.
Yeah.
Hopefully your Fridays are shorter than my Friday. Sorry, I'm wearing scrubs, but I'm teaching anatomy lab in the afternoon. It's about me up. The slides that I have up right now are mostly a continuation of the ones that we didn't finish going through on Wednesday. I added a couple more kind of at the end, so you don't really need to pull up new stuff right now, but I want to make sure that you have what I'm showing you. We're going to begin by thinking about that paper that you guys looked over for homework. So why don't you dredge those out, dredge your memory of those out, and take a couple minutes to review with the people at your table and kind of discuss what you found. So remember, we were trying to figure out what that paper is about, what their overall evidence is. That's pretty much it. Short, but we're looking for just some key things so that we get a sense of what these ancient geology papers kind of look like.
I said the most topic. This is the provided evidence of. And then I just like looked through the data that they collected.
There's a lot of numbers.
Yeah, I was not getting into all of it.
There's no error on that now. I'm not calling nothing. Yeah. This was. Oh. So like, they were saying that they did like, I guess their standard deviation are 1.9 over 7 different. So I guess it was. Just to reinforce their evidence so long that. It was just that they were right, but it doesn't. Yeah.
I'm curious, did anybody try redoing their math?
Well,
I only asked because I used that little basic equation that we used the other day, and I tried to redo them. I think you need a more complicated equation. I didn't get it to work, so I was wondering if any of you dug through and got it to work. Yeah. Okay, so basic point. So how about this table give me one basic point from this paper that they were looking at?
Like, sorry, say again.
Just any like one major thing that they were looking at in this paper. I heard both tables talking.
essentially, like, there's evidence showing that Earth is even older than they initially thought, and that's what the study was all about.
Okay, yeah, so a big part of it was about the age of the Earth. Did anybody catch a second sort of major topic that they tried to throw in there, or that they did throw in there? There was magnetic source stuff. And the other thing that I thought was interesting on here was the bit about water. Did you guys see that kind of at the end? So where they were looking at those different tables, they had the uranium decay, but they also looked at that oxygen component. So I want to talk a little bit about why they did that before we move into our next bit. because I think in the paper, they assume you know why. This is the form of a letter rather than the like whole big research article. So bonus is that it's short, but like the bad side is that real dense. Okay. So here was the here's the slide off the paper that we were looking for. We were looking for some major topics. We saw a bunch of their data on the aging. Then we saw data on different isotopes and some of their sort of trace minerals that they were finding. So just a slide of where they were looking. So they were looking here in Jack Hills in Australia, but we have other places where we can find really old rocks. So if you consult some old sources, they may be referring to one of these other locations. They were written before they found the specific site. We know that actually one of the places where it's a pretty good place to look for old rocks is kind of near us. Okay. So one of the things that's helpful to know, what was that? Okay. So a lot of these say nice on them. That's how you say the name of this rock. This is a type of rock that includes lots of zircons. And you can kind of think about it as like layered granite. I am not a geologist, so we're going to be a little hand wavy about that. But when you're looking for evidence of whether there was water on the planet, realizing that zircons form in granite, and that's where you're finding them, that's the type of rock essentially you're looking at when you're finding these really old zircons, it's helpful. And that's because water changes the temperature of things. So specifically, when there's a watery environment, an aqueous environment on the Earth, that lowers the melting point for a bunch of materials, so our minerals, things that are cooling into our rocks. So the formation initially that we were looking at, so this is right where you're talking about the magma, right? So the crustal magma coming up and cooling is what's forming these old rocks, essentially. So zircons form when granite forms at these lower temperatures. It's really common. You can make granite in some other ways, and stuff is really hot, but then you don't really end up with zircon in it. So that's kind of a sign that there was a specific type of cooling to the magma, which is why they use that partially as evidence for water. So the presence of the zircons in these sort of granite granules. People do partially use granite as a kind of proxy for temperature record for really ancient times. So here's just a picture of how they could do that. I would not be able to look at these different cylinders of granite and tell you what temperature they cooled at, but that is a field of study apparently. Okay. But the oxygen isotopes that they were looking at in this paper also relate to this cooling of the magma into granite in a watery environment. So a lot of times when we're looking at sediments, rocks, minerals to study the ancient past, we're looking at isotopes. So we talked about radioactive decay when we were looking at the uranium and the lead, but we'll also look at like proportions of isotopes present. So in our water cycle, part of what happens is that you have multiple isotopes of oxygen in the water in, say, the ocean or lake. So typically what we're looking at is oxygen 16 and oxygen 18. So they're there in the ocean. The lighter water evaporates more easily. So it kind of goes up into the clouds, evaporates more easily, but when rain happens, the heavy water snows or rains down more readily. So this is part of why you can kind of like look at changes in these ratios to figure out like whether there was a water cycle going on, because based on more complicated versions of this cycle, you can predict kind of the exact numbers. I think they actually have like, you know how there are standards for various things, like there's a kilogram, right? I think they have a vial of seawater in a museum in Vienna that like is the seawater. So different types of water in different places like have very specific kind of standards that we expect for kind of our baseline of these isotopes. So that's kind of part of what they were talking about in that last section of the paper in that table. So that partially matters because we're setting the stage for the beginning of life. So we're gonna talk about that briefly. Or not so briefly. And go ahead and discuss again at your tables what you remember about our basic requirements for or definitions of life. And honestly, based on our second day where you talked about some of you brought in talking about the chemical evolution of life, that's maybe a very brief discussion today, but go ahead and talk about what you remember about what we need, what are requirements for life on Earth. Okay. I hear a lot of talking about sort of modern life requirements. But when we're starting out the basics of life, we want to know a little bit about like just like the very, very tiny building blocks. So also on your slides, when you have a blank one, often it's going to just mean that I put a white box that you can delete on top of stuff so that we could discuss first. So know that for later, you can go into the slides and delete things. So what we hopefully realize about like the very, very basics of our very basic forms of life, like cells, right? So life is cellular and self-replicating, right? So we got full cells, those are our building blocks. We have unicellular organisms and we have multicellular organisms. Each of these cells is made up of organic macromolecules. So we have carbohydrates, our sugars, we have lipids, especially forming up part of our cellular membrane, and phospholipids. We have proteins that take all kinds of different functions in the cell, make up structures, make up our enzymes, work on our metabolic pathways. And we have nucleic acids, which form our genetic code, basically, our genetic code and how we read that genetic code to produce proteins. Now, the first bullet point, I don't know how much you've talked about before. Have you talked about like why life is based on carbon before? In any of your classes? A little bit. Okay. I'll go through real quick. Okay, so the carbon makes a difference here. Because what we have to do first is we have to form our molecular building block if we want to get to a cell. First, we got to figure out where we're going to get all these macromolecules from in the first place. Then we're going to have to make them big enough, right? It's not going to help us too much if we have like one amino acid. We know that modern life, we have 20 amino acids and then big long chains that hold up into big complicated proteins. So we're going to have to figure out how we can actually construct complex molecules from our molecular building blocks. And then to get to a cell, in addition to having these macromolecules, we're going to have to wrap that all up in a nice little baggie, keep it condensed, and end up with something that has self-replicating cycles. So we need metabolism to happen, and we need the cells to replicate also. So we're going to need some kind of genetic code. We're going to need some mechanism of copying the cell. So this is really hard to look at in the ancient past. So the various theories are, they've tried to conduct experiments on these, but there are very many kind of questions left for us when we're trying to figure out exactly how did this happen? How could this happen? But we have tried some cool things. So a first thing to note, that sometimes when people are talking about aliens, they'll suggest, what if they're not carbon-based, right? What if we're actually looking for something else? So all of our organic macromolecules are based around carbon, but theoretically, sometimes people suggest that maybe alien life could be based around silicon because it would have some similar properties. So we're going to talk first about like why carbon, why that's the basis for our life forms anyway. Okay. So when we're trying to construct macromolecules that do a lot of different things, we want a lot of different molecules of a lot of different shapes, because if every molecule is just like the same Lego block over and over again, you don't end up with much variation. Doesn't leave you room for evolution, doesn't leave you room for new invention, essentially. Okay. But fortunately, carbon has forms 4 bonds, and it allows for 3-dimensional molecules, basically, because of the shape that those bonds take. So we can see that both carbon and silicon form this sort of tetrahedral shape with their 4 bonds. Okay. So when we think about that 3-dimensionality, carbon and silicon are kind of equal. Most of our rocks, or many of our rocks, are actually silicon-based rocks. They're also 3-dimensional, right? So here you see the tetrahedron and how that works in our zircons, which we've talked about, but other things like quartz is probably the next most familiar thing on this list, but these are all other types of rock, essentially here. We're showing that sort of crystal structure based on the tetrahedral shape. But carbon has some benefits for life itself. Yeah. So a helpful thing when we're looking at carbon is carbon bonds pretty easily to itself. So we can make carbon-carbon bonds about as easily as we can make carbon-oxygen bonds, et cetera, et cetera. So this means that instead of ending up with kind of separate chunks of carbon surrounded by 4 bonds, we regularly end up with carbon-carbon backbones for things. So if you think about like, I've been looking at lipids in A&P recently. So, you know, if you think about like a fatty acid, right? So one of our lipids, right? Our carbon backbone there is super obvious, right? Just at each of the points. So that carbon-carbon backbone is helpful for building macromolecules because then you can make your chain different lengths. And we can also swirl it into a ring structure, make different shapes that way. Carbon, when it forms its bonds, is also basically the bonds are kind of more flexible. And to understand that, we can think about kind of the structure of the atom. So basically, silicon is bigger, it's heavier. So if you're looking on the periodic table, right, it's in this row below. Okay. So while it can make 4 bonds, the sort of body of that molecule, the nucleus of that atom, I should say, it takes up so much space that those 4 bonds can't really stretch that far, essentially. Whereas carbon, much smaller, can make those bonds at sort of more differing angles, make them farther apart, basically not break as easily. So we have more options for the shapes that we can make there. Another thing that's cool about carbon is that at the same temperature and pressure, so like the temperature and pressure in this room, you have some molecules based on carbon that are solid, some that are liquid, some that are a gas. And that's kind of special. That means that we can have carbon-based molecules that are transitioning states pretty easily in the same environment. Okay. And that's helpful when we're thinking about metabolism. Well, metabolism is kind of a really important thing that we think about as we think about the very beginning of life. We need energy in order to do anything at all. So carbon is also able to form molecules that are soluble or molecules that are insoluble. And it can also have multiple valence or oxidation states, which allows for energy transfer. So basically we have a lot of options of what to do with our carbon. And just a note, because I don't always remember a ton of chemistry. So that last bit, basically, when we're looking at those oxidation states on the carbon, we're talking about like how easily are they holding on to electrons? Are they giving away? So as we think about in the future things like cellular respiration, breaking down glucose is technically glucose oxidation. Glucose is a carbon-based molecule. We can partially do that because of this ability of carbon to be at multiple oxidation states. Sorry, turning on my do not disturb. All right, so that's why we're starting with carbon. Now we're going to try and build molecules. So we don't really see direct evidence of life on Earth until about 3.5 billion years ago is where we can see something we can actually see directly and we're like, oh yeah, that looks like something I recognize. But we have to come up with, or we want to come up with an idea about how we got there to that visible life. So starting back in the 1920s, this guy Oparin and Haldane, Oberker, I guess, came up with a hypothesis of abiogenesis. So basically that life came from something that was not living. So our building block for life are amino acids, which would eventually be our proteins, started as something that was not alive and then combined to become more complex. Now, that does seem a little crazy occasionally, or it did for sure to people in the 1920s. But we actually have found this process happening around us in space. So we found actually amino acids in comets. So here's one, I think this, does this have a year on it? This is a relatively recent comet that they found glycine in. And they've also recently found tryptophan, another amino acid, in a star forming region 1,000 light years away from Earth. And they can tell based on basically the spectra of the light that's being emitted from that star forming region, even from this far away. So the formation of amino acids does seem possible because we've been observing it. And that's just 2 of our 20 amino acids, but it's a little bit of support for the idea that this could happen. Also, kind of fodder for the idea that life could have extraterrestrial origins, that part of life or part of the components of life could have come from, say, a comet or a meteorite from somewhere else. I saw a paper, a group at Max Planck is working on thinking more about protein formation. And so they've been modeling like comets hitting Earth, and part of that impact apparently they think helps with the formation of larger proteins as well. I didn't pull that out for us because it wasn't fully delineated, but it's an ongoing area of research basically. So then as we progress forward into the 20th century, there's a really famous experiment called the Miller-Urey experiment. So basically what they did to figure out whether they could create some of these macromolecules is they decided to create a closed container that had the components that they thought were present on the early Earth. So here we have their system with a bunch of tubes full of water here. We have gases over here in what they thought were the same proportions that were present around 3.5 or 4 billion years ago. So what they did was they heated up the water to be their sort of fake ocean, right? So this is why it's important to actually establish that there would have been water, otherwise this is not very helpful for us. So they heat up the water, we got water vapor going through. And in this other chamber, they had the gases that they think were present in the primitive atmosphere. So you see oxygen isn't on there because we're not gonna have a ton of oxygen gas in the atmosphere until we actually have life producing that oxygen gas. But we have some other things that we think are in the primitive atmosphere at this point. Then they applied electrical charges to simulate lightning, condensed down what came out of this sphere, and sampled what they ended up with. Now, the first time they tried this, I believe it did not work. And then they realized that they had the portion of gases wrong. They tried it again, and they found that when they adjusted the ratio of the gases in the primitive atmosphere, they did get many organic molecules forming just by this kind of simple experiment. So they did end up with a bunch of amino acids. They ended up with some sugars. They even ended up with lipids. So that's another little piece of they tried to do to show that this is theoretically potentially possible to form these molecules, the components of life from non-living things. Another idea about where life may have come from rather than a lightning strike in the atmosphere is that actually deep in the ocean is another potential site for the formation of these first molecules. And so deep on the ocean floor, we have some hydrothermal vents. So they're venting up a lot of heat, a lot of gases out of the ocean floor, so from deeper down. As they do this, they end up creating all sorts of gradients between the colder ocean water surrounding and this water that they're heating up and pumping full of all sorts of minerals out of their stacks, basically. So think of them as a little volcanoes there. So one of the things that happens in a certain type of hydrothermal vent is that they create a gradient of protons here in that difference between the different parts of the water that they're spewing into. So when we think about protons and we think about gradients of protons, we can again think about kind of what happens in the cell during metabolism, right? When we create that gradient across the inner membrane in our mitochondria, right? We use that eventually as our energy for creating ATP in a cell. So this is an idea that suggests that maybe this could have been the source of energy for chemical reactions to happen as life was forming. There are also all sorts of hydroxide ions, minerals, other trace components coming out here. So it would also give them basically different atoms that they could combine into these chemical reactions. And they already form methane and acetate on their own, which we can see down here. So those are some of the things that we can see them forming right now in real time. So that's helpful to realize that they're making these kind of little building blocks right now. And we can see that happening abiotically. There's also a temperature gradient could potentially be helpful. And the little pipes inside the hydrothermal vents, the ones that they think could have been the start of life, are something called serpentine hydrothermal vents. So essentially think of like a narrow little winding tube going up. So some people have suggested that that might have helped early life kind of getting stuck in corners of the vent so that you end up with the few building block molecules that are being created kind of getting stuck and clumped together in regions, because that's actually another kind of problem we have when we try to think about how life forms, right? Like it doesn't help if you just form one amino acid every, even if it's like every 10 minutes, right? It's all gonna kind of spread out. You're never gonna end up with a lot of amino acids right next to each other if you're forming them infrequently. So another problem we have to contend with when we're trying to figure out how you actually end up with a cell is how to do clump all that stuff together. But even if we can get all of our amino acids, we have to make a larger macromolecule. We want to bond things together into things like proteins. That is also still an active area of research, how that happens. So one theory is that it's in kind of corners of hydrothermal vents. Another theory is that it might have to do with water evaporation. So if you think about these building block molecules forming in the ocean, whether it's from a lightning strike or whether it's in hydrothermal vent, you'd end up with them suspended kind of in water. Water forms droplets and droplets dry up. So if you end up with a droplet of that somewhere on land, as the water evaporates out, things like amino acids would still stay in that droplet that's getting smaller and smaller and smaller and would condense together. Now that ends up with them in the right spot, but it doesn't end up with them in quite the right orientation. So a tweak to this theory to explain how it could be possible that we end up with our amino acids in the right sort of orientation to form our more complex patterns would be that if we have our water droplet evaporating on a piece of clay, clay kind of has pockets and could act kind of like an enzyme, right? So the clay could physically hold things in place while we kind of wait for that reaction to happen. So there are a lot of big question marks when we're this far back in the past. Big question mark on forming these molecules. Creating our first cell, once we have those macromolecules, honestly might be a little bit easier because of the properties of phospholipids. So phospholipid properties already kind of form, based on their chemical properties, form cell-like structures or cell membrane-like structures if you drop them in water. Right? They're amphipathic, so we have a polar hydrophilic head and our non-polar fatty acid tail. So they're going to orient relative to water. And one of the structures that they naturally form as those heads are moving towards water, tails are moving away, is something called a liposome, which is just a lipid bilayer sphere. So we can form little micelles with all the tails in the center. But if you end up with some water inside, everything's going to reorient so that we always have those hydrophilic heads facing water inside and outside our sphere. But that part actually isn't all that hard. So once we get our macromolecules, the right ones, in a concentrated region, would be relatively easy to get a phospholipid layer to form around them. An alternate hypothesis about how all this works is that we don't need to start with proteins and amino acids. Because even if we have our proteins and amino acids, then we still have to figure out how we get to the self-replicating part, right? We still have to figure out, okay, so how do you end up with DNA that is forming RNA and protein and replicating itself into more DNA? So a relatively popular hypothesis is that actually we could just start with RNA. So this is called the RNA world hypothesis. So RNA can form many different types of structures. So here's the idea, right? We do have to figure out how to get our first RNA to form, right? So that would be a similar problem to our amino acid formation problem, although you don't need to make 20 building blocks of RNA, right? We have fewer types, so that's a little simpler. Okay. Now, RNA can act as a part of genetic code. So potentially it could self-replicate because can form both that kind of original code for a molecule and also using ribozymes, which are kind of crumpled up balls of RNA, can kind of act as an enzyme. So you might have two components there that work together. So the ribosome as the genetic code and also, sorry, the RNA as the genetic code, and then that sort of proto-ribosome as our enzyme and potentially even our protein builder later. So we self-replicate our RNA, and then we would loop over here. After that's going on, we might end up with that RNA catalyzing protein synthesis based on that RNA as well. So that would be the basics for this RNA world hypothesis. Then we'd have time continue on and membranes form around this kind of self-replicating chemical pathway that we have going on with RNA. So you could have this going on not in a little baggie and then eventually have some lipids, some phospholipids trap a little bit of this cycle and keep it contained within a membrane. And then you end up with kind of a protocell, right? So you'd have the RNA doing this within a membrane. So under this theory, we would then have RNA as our first genetic code and DNA form kind of like as an evolution from RNA. So that would be step 5, getting to DNA as our master template and separating out our proteins there. So however you slice it, we have lots and lots of questions to answer when it comes to understanding how we ended up with our first cell. It's hard to look because it's way, way in the past. I am going to cut out a picture sheet of some molecules related to this RNA world hypothesis. Actually, I'm just curious. So everybody, do you mind putting up a hand and put up a finger 1 to 5 on your comfort with chemistry? Like 1 being not and 5 being I am an amazing chemist. I'm just curious. I'm probably like, no, I'm not that. Okay, cool. I just want to know where we are at sort of details of chemistry. So what I'd like you to do on this sheet of molecules is I want you to go through and try and identify them, any of these that you recognize. And then I want you to look at them and think about why some scientists look at these molecules that we have in our body now today and think that they might be evidence or like remnants of this RNA world. Right? They're not evidence, they're not like proving anything, but if this RNA world hypothesis were true, then these molecules are used as signs that some of this has stuck around, even though we now have DNA and much more complicated and efficient systems. Oh, you can do it together. Yeah.
These are all.
Sometimes I end up with like a lot of biochem majors in class. I'm like, oh, guys, yeah, you can explain the chemistry. So I'm just curious. And if you can't identify them, still think about just looking at the shapes, why this might be related to RNA.
By studying all of them. Yeah. This is the report of the Colonial trial. This is.
Yeah, I know, you can come out.
Don't know about. I would believe so, yeah. Which one? Five,
Yeah, that would be.
Yeah. One of them has the. This has an iron. But I think the iron actually. I think this is the. Come on.
Very exciting.
Why is the vitamin? You think I'm
So you're doing this right now, we're gonna work. We're on it now.
Okay, so while you're looking at these, also try and find the piece of all these molecules that is the same. Okay. So as y'all are looking at this, I'll pull up on the slides the identifications of the molecules so it doesn't drive you crazy. But from looking at these molecules and looking, hopefully some similarities you can see there, do you see why scientists think that this could be related to an RNA world? What do you consistently see in these molecules? Do you see anything? There's definitely oxygen.
5-member like base ring with the oxygen on top.
So a reminder before we look through. Okay. So we remember, so ribonucleotides, right, are going to have the ribose sugar, they're going to have a phosphate group, and then they have a nitrogenous base. So when we look back at these molecules that we see today, So this first one, here's some people say, so this is acetyl-CoA. So this is an important part of our metabolism today. So a coenzyme that is important to our production of ATP. Okay, so we can see in there, right, we have our, which of those phosphate groups I want to circle, I don't know. I'm going to grab this one at the bottom though. Right, so we have this sort of basic structure. It kind of does resemble our ribonucleotide, essentially. And then we click through. So this one was hopefully familiar as well. So this is ATP. All right, so we can again find that nucleotide hanging out here. This one for sure looks like ribose because we have both of those OH groups at the bottom. Okay, so we can see that in there. So both of these two molecules are really, really involved in our production of cellular energy. And they both do look kind of RNA based, right? They do look like modifications of a ribonucleotide. Going through the rest of these, we can see similar sort of things as well. Clear all these drawings, right? So if we're looking here, this is vitamin B12, okay? So also involved in the production of some important cellular components. So we can see that ribonucleotide sort of down here. We also have NAD, right? So again, we can see those nucleotide structures kind of glued together. This is an important electron carrier in metabolism. And the other one we pulled out was cyclic AMP. So this is part of important cell signaling pathways inside cells as well. So cyclic because it's got a circle, but again, we got sugar, we got a phosphate group, we have our nitrogen base. So scientists suggest that if the RNA world hypothesis were true, which they're still trying to figure out all the bits of that, right? Can't prove the idea. But if it is true, then this might be evidence left over from that previous form of life, essentially. These molecules based on nucleotides that are still really important for metabolism and basically our energy pathway. It's a very, very fundamental part of what makes something living. Right? So you can imagine why that might be part of where you end up with these molecules kind of sticking around for longer. Okay, there are more RNA-derived compounds that are still present in life right now. So pulled out some here. The ones I pulled up on this worksheet are the ones I just imagined you might have talked about in your previous classes, even if you haven't seen them drawn out in a while. Okay, so basic idea in RNA world, you had RNA genetic code, crumpled up RNA, helped read that to make more RNA. Eventually that RNA started pulling in amino acids. So we started to see protein formation and we ended up with this RNA protein cycle. And that eventually out of that process, later on, new life emerged that used the RNA to form proteins, but ended up with double-stranded DNA as the basis of the genetic code. So the central dogma in cell biology is that idea that we go from DNA to RNA to proteins. Now, in order to assemble a full picture of how this would have worked, does take and would take a lot of different areas of research to kind of figure out how this is possible and ideally try to test it. So here are some of the questions and things we would need to understand in order to get a full picture of that RNA world. That's what we're doing. Okay. So we got to form our macromolecules out of our building block molecules. That is difficult and difficult to understand. But once we have our simplest form of existing life, we're going to get into things we can start to see in the fossil record, which is exciting. Oops. I guess that's the answer for you. Okay, so does everybody sort of recognize what this will be? So our simplest form of existing life are prokaryotes. So there are unicellular organisms with these common components. So this is a prokaryotic cell over here. So what we can see is that we have a plasma membrane on the outside. Okay. We have cytoplasm inside. We have DNA in this sort of nucleoid region, but not contained within a membrane. And we have some ribosomes. This particular prokaryotic diagram has more things, but those are our basics when we're thinking about our prokaryotes. So they're simpler than the cells in our own body. They are eukaryotes. The prokaryotes are unicellular. So we expect that the first forms of life were unicellular organisms, probably similar to prokaryotes because that's easier and simpler to form. They're also much smaller than eukaryotes. They lack a nucleus and in general lack all sorts of organelles, lack anything that's basically internal membrane. So we only have to form the one membrane around our various cellular components. So eventually, we expect that we're going to go from some type of prokaryotic ancestor to our more complicated eukaryotes we have around in the modern day. We do still have prokaryotes as well, but we think that the eukaryotes came from prokaryotes. So that would look something like this. You can see my amazing drawing. So our little eukaryotic cell and a prokaryotic cell like bacteria. And then our little bacteria ends up inside the eukaryote, and ta-da, we have organelles. Okay, cool. So that's endosymbiotic theory, but we're not moving past prokaryotes today. So what we are going to finish on is thinking about cyanobacteria and the first direct evidence we can actually see in the geologic record of life. Okay. So cyanobacteria are prokaryotes. They're also known as blue-green algae. And a cool thing about them is that they're capable of photosynthesis. So we're not going to go through that whole photosynthetic pathway, but I wanted to take a look at kind of a comparison of a plant cell versus cyanobacteria, because typically when we think about photosynthesis, we think about plants, or at least I do. So you can see how in the cyanobacteria, we have some of those same pathways going on, just kind of inside the little tiny bacterium, okay, that are going on in a plant cell inside the chloroplast. Right? So you can think about the chloroplast as maybe an ancient remnant of a cyanobacterium that got swallowed by something else. Okay, cool. So what we can see, which is very fun, very exciting, and I have some. So what we can see in the fossil record are which, but stromatolites are our first evidence of sort of visible life on Earth. So what stromatolites are basically microbial mats that can fossilize after they form these long, tall structures. So when you're looking at stromatolites, you're looking for stripes essentially within the rock. So you can see these alternating dark and light layers where the photosynthetic cyanobacteria would have grown. So we can date them back to about 3.5 billion years ago. So if we're thinking about why they look that way, helpful to understand why a stromatolite grows that way, to understand why we think we can identify this in a rock. So when we have these cyanobacteria, they are unicellular, but they like to grow essentially in colonies, right? They grow together with other unicellular cyanobacteria or blue-green algae. So they end up forming these basically mats or film on the water. So that mat starts to create a kind of sticky film on the big group of cyanobacteria. And then sediment gets trapped in that mucus layer. Now, these cyanobacteria, these microbial mats are typically in or on the ocean or tidal pools or something. So they're going to keep growing up because they're going to keep growing towards the sun. They don't want to end up under the mud that's collecting. So you're going to end up with another new living layer on top of that first layer that kind of congeals. Okay? As this happens, calcium carbonate gets kind of back into the old layers and fills in and turns it into limestone. So these layers grow and grow and grow until we end up with kind of towering stromatolites. And this process repeats over and over. So this is the internal structure, but I should point out on the next slide. Here we go all the way back to the first slide I pulled up. Not the fastest way to do that. Okay. So we do see stromatolites now today in real life. So these are some in Australia, right? So we know how they're formed, not because we're guessing, because we can literally see it. Okay, so these are stromatolites that you could potentially go visit, modern ones. Back to our ancient stromatolites. Okay. So when we're looking at those stromatolites, buried in these really, really old rocks, we can see these layers and these shapes that we know look like stromatolites based on looking at modern living stromatolites. Everybody get a look at the stromatolite? See if James can find one. All right. So we can find those in our really, really old rocks. Also in Australia, funny enough, just like those jackpills zircons. Okay. The final bit of sort of trace evidence of life are our carbon isotopes. So we're not going to do complicated chemistry with this, but just breaking down isotope stuff. So carbon comes in multiple isotopes, so like multiple different versions. So carbon 12, 12 being our nuclear number, has 6 protons and 6 neutrons. So have carbon 13, 6 protons and 7 neutrons. There's also carbon 14. We'll talk about it when we get recent enough that carbon 14 is relevant, but in the very distant past, we're not thinking about carbon 14, just carbon 12 and carbon 13. And it is true that life prefers carbon 12. So some metabolic processes favor using one isotope of carbon over the other. So when we look at really, really old rocks, at the ratios of carbon 12 to carbon 13, we can start to imagine whether that carbon came from a biological source or whether it came from just non-biological abiotic processes. And we start to see lighter carbon, so more carbon 12 in it, which is typical of life now, around 3.5 billion years ago as well. Although they pull up a paper that looks ominously very like the paper you read for homework that said they found some potentially biogenic carbon based on these carbon signatures all the way back in a 4.1 billion year old zircon. So there is a lot of uncertainty in how many billion years ago exactly we want to go when we're trying to figure out when life started. I'm not going to make you read that paper. That's where we're gonna end today. So no homework. Enjoy your reading. That was heavy, right? Yeah. I like it. This one says it's from the Iron Range, so potentially,
Oh, yeah.
maybe we can find something like this someday.
I
You found one?
I brought my.
Smells like crayon.
Yeah. Did you?
So,